in front of this August gathering. So welcome to all for my presentation on nanotheranostics in drug targeting. So today I'm going to present my talk on the nanotheranostics in drug targeting. And while presenting, I will cite some of my research outputs as an example in drug targeting. So using the nano technology. So the objective of my presentation is to have a discussion on nanotheranostics and its relevance in utilization of site-specific control and sustained drug delivery. To highlight some of novel approaches and its research potential in designing of such nanotheranostics for therapeutic targeting in modern era. And to have a discussion on the research outcomes at present scenario on its therapeutic applications. Coming to the discussion about the nanotechnology versus nanomedicines. What is nanotechnology? This nanotechnology defined as the creation of functional devices in the nanometer ranges and the exploration of these unique properties of these devices in various fields. So nanomedicine is the term. Nanomedicine refers to the application of nanotechnology for diagnosis and treatment of disease. It deals with the interactions of nanomaterials. It may be its surfaces or particles or analytical nano devices with living human material, cells or tissue or body fluids. It is an extremely large field ranging from the in vivo and in vitro diagnostics to therapy, including targeted delivery and regenerative medicines. Now, nanotheranostics, what is it? The nanotheranostics is the integration of both the diagnostic and therapeutic function in one system using the benefits of nanotechnology, and it's extremely attractive for personalized medicine because treating cancer is not a one-size-fits-all scenario. It's required therapy to be adapted to the patient's specific biomolecules. Now, nanotheranostics, why? To optimize the drug's therapeutic effect, convenience in administration, and minimize dose size and toxicity concerns. To overcome stability problems of drugs in vitro, in vivo, before, during, and after administration. To precise targeted and drug delivery. Now, if you look at this picture, in one platform, I am showing comparing the two administration models. One is conventional therapy and one is targeted drug delivery. If you see this picture, in conventional therapy, same injection mode drug is administered to the body. Whether it is required or not, it is distributed throughout the body and release the drugs throughout the body. Among that, a fraction of the drugs may be reaching to the site of action if it is desired to be released at the near to lungs or so, maybe tumor targeted and all. So a rest is wastage and giving side effects to the other organs where the action is not desired. But in case of this targeted mode drug delivery system, you see, same we are administering through the injectable modes, but drug distributed throughout the body, but not released other sites or organs, except it reaches to the particular target site. And you see it is concentrating on the particular site. It is accumulating and concentrating or attaching to the particular site of effect, and then started releasing the drugs, and which can be imagined from outside by the radio imaging technique, what is happening, how drug is releasing, and at what rate it is healing the cells or say organs or the particular disease conditions. It is observable from the outside. And so in one system, it provides multifunctionality in drug targeting, delivery, and reporting. It provides improved therapeutic index because side effect is minimized and provides lower toxicity and side effect. And target site-specific drug delivery by biosensing. So we can conjugate it with the biomarkers to identify the target cell or say tissue or say target organ to attach the system and release the drug material there, and can deliver multiple drugs directly to the tumor sites. Now you see multifunctionality in nanoparticle-based therapies. You see here targeting, delivery, release, and reporting. You see in this nanoparticle model, here we conjugated several. There are maybe one or two drug molecules loaded inside and attached with the PEG, maybe surface modifier or adsorbents. Then the target moiety, maybe we are conjugating with the biomarkers or the monoclonal antibodies to identify the target site, and then permeation enhancer to penetrate. So in this, if you see this targeted molecule or say nanotechnology based formulation, when coming to the cell surfaces, it adsorbs here. Then with the endocytosis, it goes inside. Then the endocytosis mechanism, phagocytosis mechanism, the cell surface is, the coated surface may be released, the destroyed, and the drug released inside may give therapeutic effect then and there, or kill the cells, or maybe heal the cells, and then it goes out to the normal phagocytosis process. So now if you see these animated techniques, how it affects, first your system targets the cell targets, then entry by the, so this targeting moieties may be antibodies, DNA or RNA, or peptide sequence, or a ligand or aptamer, which helps in identify the target cells and attach to the cell surface, then started penetrating, intercellular penetrating, then biomolecular sensing by the biosensors, then drug delivery to the site, then effectiveness. Now, I am going to show you certain animated video presentation. If you see, doxorubicin loaded drug delivery in breast cancer targeting. So it's a animated techniques by the company. So United States, it's found that the cancer is enormously increasing. And this nanoparticle based drug delivery system, where anti-cancer drug is loaded with, if doxorubicin is taken as a model drug, and there are several oncomarkers is conjugated with the drug molecules at nano level. And this molecule conjugated nanoparticle capsules, this is an animated model. So it conjugated with DNA origami capsules, immune factors, and molecular binding sites. So while delivering this through the blood circulation, the oncomarker will identify the tumor cell. Then PG-based materials or say polymers will help in absorbing to the cell surfaces. Then penetration enhancer helps in penetrating these nanocapsules to the cell. Now, these nanocapsules enter to the tumor cell. And then this outer surface, the polymers will get dissolved and start to release the drugs then and there. Now drug delivered inside the cell and kills the tumor cell. So it is the targeting to the breast cancer cells. So doxorubicin now effective to kill the cells. Now the merit, if you consider, first nanoparticles, because of their small size, can penetrate through smaller capillaries, which could allow efficient drug accumulation at the target site. It has been found that uptake of 100 nanometer size particles by the intestinal tissue was 15 to 250 fold higher compared to the large size microparticles. And second is the use of biodegradable materials such as natural or synthetic polymers for nanoparticle preparation can allow sustained drug release within the target site over a period of days or of even weeks. Third is the nanoparticle surface can be modified to alter biodistribution of the drugs or can be conjugated to a ligand to achieve the target specific drug delivery. Now, techniques used for preparation of nanoparticle system includes emulsification, solvent evaporation method, spray drying or congealing method, supercritical fluid technology method, parallel ball milling technology for the production of drug nanocrystal, or high pressure homogenization technique, air suspension techniques, and cross-linking or surface polymerization technique. Among them, 1-2 examples I will cite with some of my experimental outputs. How is it? Now a common commercial example I am giving: polymer protein stabilized nanoparticle preparation through emulsification solvent evaporation method. Here 2 phases we have taken. One is paclitaxel is taken as a model drug, and methylene chloride as an organic solvent, because paclitaxel insoluble in water, so methylene chloride taken as a organic phase for the distribution of the drug in it. Now, the albumin coated nanoparticle we are going to prepare, and albumin is a rate controlling membrane, can be acted as polymer. Now, albumin dissolved in water, aqueous phase, then organic phase added to the water phase with mild homogenization to form the crude emulsion, then high pressure homogenization subsequently, and the cycles and the speed determine the fine emulsification and the particle size. Now, after this rotary evaporation, methylene chloride can be removed, and with lyophilization technique, because it is an albumin loaded nanoparticle, so stability is very important, and temperature we have to control for its stability. So by lyophilization technique, we can remove the water vapor, and it can be dried and kept for the longer period. So it has been found that with this method, paclitaxel nanoparticles we got 160 to 220 nanometers. Now, with this technology, we formulated the lacidipine nano suspension using nanopure technique, because the rationality behind with this preparation of the nano pure suspension, because the lacidipine is, is a cardiovascular drug and used for the lowering the hypertension. So it was poorly water soluble drug, and one of the critical problems associated with poorly soluble drug is poor bioavailability and erratic absorption. So this paper published in Albanian Journal of Pharmaceutical Sciences in 2014. Now, this is the formula we utilize, where we tried, there are different formulations you will find. We tried the lacidipine nano suspension formulation using different polymers like hydroxypropyl cellulose, polyethylene glycol, and polyvinyl pyrrolene. Among these three, and we later this nano suspension absorbed to a base material in granular form, and then we converted into tablet form for in vitro in vivo stability and utilized further. Now, in the nano suspension, when we prepared, we found that this HPC, the hydroxypropyl cellulose, is particularly effective in nano grinding and producing very fine small particle size up to 200 nanometer ranges. But others, PG to 20,000 and the polyvinyl pyrrolene was not found that suitable to form the nano level of particle size. So we, among these formulations, the stable formulation formulated with the hydroxypropyl cellulose taken into further study. Just because of time shortage, I will give the comparative statement, what the result outcome. You see the reference drug, lacidipine, and the nano formulated lacidipine suspension. So we have in vivo studies done in the rabbits, and it has found that, you see from the graph, we can compare 6 times in the 1 hour, within 1 hour, 6 times concentration in nanogram per ml, 6-fold increase in bioavailability in the plasma. So you see the nanotechnology approach, how it increases the bioavailability. Another example I am going to give is formulation and characterization of in vitro in antitubercular activity and cytotoxicity study of solid lipid nanoparticles of isoniazide. As you know, this is a global problem, and the drug-resistant tuberculosis is increasing day by day. So we tried in the nano formulation to deliver precise delivery with sustained delivery of the isoniazide. So this is published in the Journal of Nanobiomedicine and Engineering in 2018. And these isoniazide loaded solid lipid nanoparticles were successfully prepared by high pressure homogenization following ultrasonication technique and optimized using 2 to the power 3 full factorial design. These isoniazids loaded solid lipid nanoparticles were prepared using the stearic acid, poloxamer, and the soy lecithin as the base material for the solid lipid nanoparticle vesicles. And then isoniazid was loaded in that. So this isoniazid loaded solid lipid nanoparticles was characterized for the particle size. Just I am going to discuss the outcomes. Particle zeta potential and percentage entrapment efficiency and the cumulative drug release study. So physicochemical properties were investigated using transmission electron microscopy, differential scanning calorimetry, and X-ray diffraction techniques and FTIR. The average particle size, zeta potential, and percentage entrapment efficiency of the optimized formulation was found to be 167.1 nanometer, and with the zeta potential and minus 32.4 millivolt, and the percentage entrapment efficiency found to be 73.17% respectively. Now, the result, if we discuss the optimized formulation, shows cumulative drug release of 79.14% up to 36 hours. So it helps in, it justified the sustained delivery. And in vitro antitubercular assay, luciferase reporter assay is conducted in H37RB viable and resistant strain. And then cytotoxicity study, MTT assay, the mitochondrial toxicity study was conducted using the J77-4A.1 cell lines, and the isoniazid loaded solid nanoparticles were evaluated and compared with the free isoniazid. The result of LRPSS says that in the tuberculosis resistant strain showed percentage relative light unit reduction value of isoniazid solid lipid nanoparticles, and free isoniazid was found to be 99.75 and 99.89% respectively. Whereas in case of isoniazid resistant strain, it was found to be 90.27 and 90.52% respectively, conferring notable anti-tubercular activity. So as free drug, it shows the anti-tubercular activity similar to the free drug, and but you see the MTT assay reveals that the percent cell bioavailability upon exposure to the isoniazid loaded solid lipid nanoparticle were significantly higher, more than 90% than free isoniazid. It is less than 80%, confirming it is safe to be used. So these are some of our research output I discussed. Now, if you see to the market potential and some available market products, nanoparticulate formulations, Oncospar, it is prepared by the Sigma Tau Germany, acute lymphoblastic lymphoma, it is going to be utilized, and Abraxane, American Bioscience is producing for the breast cancer. Then Pegasus is the commercially available trade formulation. It used against the hepatitis C, and nano-liposomal formulation, Avelsar, liposome company, New Jersey, USA, fungal infection. Then Epaxel, Swiss serum company, used for hepatitis A. Then fullerene loaded nanoparticles, radical sponge, used for vitamin C60, cosmetic use. And Dendrimer's formulation, superfact by Quagen, lipoprotein lipase deficiency. So these are some of the commercial examples I am giving, because already these are available in the market, and it is a huge potential to work on the nanotechnology based formulation for the market. Now, toxicity, when we are formulating the nanoparticle based formulation, it is also our responsibility to look to the toxic manifestation of the formulations. So toxicity also a concern of nanostructure particles and materials. So one of article I published, bioaccumulation and toxic profiling of the nanostructure particles and materials in the open science books, unraveling safety profiles of nanoscale particles and material. So here use of nanotechnological based formulation is increasing day by day, and the nanomaterials are in wide ranges covering a broad typology of applications, from design and development of targeted drug delivery systems, manufacturing of pesticides, domestic appliances, textiles, to bioremediation engineering. So there are therefore concerns about the environmental risk of bioaccumulation related issues that may arise, particularly resulting from the application of drug-loaded nanocarriers and effect of pesticides that reaches the natural ecosystems. So it is also known that nanoparticles made up of non-degradable or slowly degradable materials may accumulate at the cellular level and hinders the enzymatic activity of the functional proteins. So the toxic manifestations may also arise due to their chemical composition and structure and surface charges, solubility and aggregation on the site of the elimination or filtration, such as the glomerular filtration of the nephron. So the mechanism particularly involved in this bioaccumulation and toxic action includes toxicity of nanoparticles due to dissolution, high penetration, high availability, and maybe affecting to the sense organs. And cellular uptake of nanoparticle is very high, and their effect on biological membranes and oxidative stress induced via nanoparticles and nanomaterial cellular uptakes. Now, examples of some cytotoxicity related research outcomes, these are not my outcomes, it is from the others research output, just I presented because toxicity also a concern, major concern, and we have to be conscious while conducting research on the nanoparticle based formulation. So among the available manufactured nanomaterials, titanium dioxide is one of the most widely used to date, and previously studies suggested that titanium dioxide nanoparticles production ranges from 7,800 to 38,000 metric tons per year in USA alone, and will be approximately 2.5 million metric tons by 2025. The fate of bioaccumulation of titanium dioxide-based nanomaterials and nanoparticles in a body microsome over a period of 17 days shows that the titanium level were the high in biofilms during the exposure period. So titanium is a heavy metal, so you see the toxicity. So bioaccumulation factors indicated that titanium dioxide-based nanoparticles and titanium dioxide-based nano tubes were largely transformed from the low to superior trophic levels. So considering the potential entries of titanium dioxide-based nanomaterials in organisms, their bioaccumulation throughout the food chain should be regarded with the great concern in terms of overall health of the natural ecosystem. So unless and until we are conscious and we are assuring the safety concern of the nanoparticle based formulation may be utilized for the therapy, it may be dangerous to the society. So we have to assure that the safe to be utilized, our formulation is safe to be utilized for the purpose. So the concluding remarks I am going to tell you, the advanced pharmaceutical technology is expected to have great impact on many areas in medical technology to change healthcare in a fundamental way, especially surgery, cancer diagnosis, cancer therapy, biosensing, molecular imaging, surgical implants, tissue engineering, and drug delivery and gene delivery. So widely, day by day, we are going to concentrate on the nanotechnology based formulation, and I am sure this nanotheranostics, where both the techniques are integrated to diagnose and release the drugs, specific delivery and precise delivery of drugs, helps in wide application in the healing the several diseases, including cancers and the tuberculosis and all. Lastly, I thanks to give my acknowledgement to Dr. Sunil Kumar Panda and Dr. Subas Chandra Dinda, who happens to be my scholars who helped in these two areas to work and give research output for particularly which I given the cited my examples during the presentation, and the Berhampur University of India, which given the facility to conduct some of the experiments, and National Institute of Research in Tuberculosis, Chennai, India, for giving the facility to conduct the MTTSA and the LRPSS. So these are some of my references which I followed and which are published. These are some of my publication and published in the journals or the book chapters and quoted here. The extract is presented inside the presentation. So thank you all for patient listening.